Okay, so the postings, for those of you who are not here, I'm just saying this again for people who listen to the recording later, that there's some articles that have been posted in EDUCAT, as well as a video link that is on how plants defend themselves, how they talk to each other, so to speak. So this is where we left off, though, in the slide set. Looking at, on this one slide, there's a lot of information getting into once the plant is detecting that something is going on, it needs to pay attention to in terms of a pathogen. Here they're showing maybe it's a piece of the pathogen, maybe it's part of the cell wall or flagella, but some part of the pathogen docks into the receptor protein, and then they're trying to show how that kicks off a signal cascade response. And so you see a lot of things happening here, lots of arrows. So we're going to work through this and kind of work through step by step some of those terms that are listed in the white box. So actually, okay, so this slide, you can see how there's question marks in certain parts of the pathway. So I put this in here just to show you that just a few years ago, this was all still like, I don't know, we don't quite know what's going on here. But this is the updated version from your textbook, and you can see that they're starting to fill in the blanks. So there's more material. This is an updated version of that slide that we just looked at, filling in the detail. So we're going to get into this idea. Again, you can see calcium as a signal transduction, the second messenger in that process. And we're going to talk about nitric oxide and some of these hormones that get ramped up. We'll talk about reactive oxygen species as well. So with the reactive oxygen species, just remember we're talking about these ideas here, that you tend to learn about reactive oxygen species as, you know, they're bad, bad, bad. And they can do a lot of damage, certainly. But I want to also impress on you that for plants, reactive oxygen species is a source of information as well. It's not just this evil thing that's going to destroy their membranes and proteins. They actually use it as a signal molecule, as a way to kind of figure out what's going on. So that's what you're seeing here in this diagram, this idea of reactive oxygen species being made, and that is a way for the plant to sense that they're faced with a challenge, whether it's some kind of abiotic idea, which is being shown on the left side of this figure, or right now we're focusing in on biotic pathogens of some sort. And so they will use that reactive oxygen species that ramps up in their cells. They'll use that as good information so that they can respond appropriately. So let's get into that a little bit with the hypersensitive response. So this has been documented for quite a while. This is something that the plant can do when a lot of the studies that they initially did with this was looking at fungal attack, when a fungal pathogen is attacking a certain part of the plant body. And they were curious to see how when the fungus starts attacking a certain section of the plant body or tissue, cells in that area start to go through programmed cell death or apoptosis, with the plant directing that. So it wasn't the fungus doing that, it was the plant doing it. And so what happens is the cells around the fungal hyphae will just start to die. And then what they see happening is those dead cells, the nutrients get siphoned away from the fungus and get sent into, you know, other plant cells or through the phloem so that it gets away from the fungus. So the more common way to say that is the cells are committing suicide. It's natural programmed death, and it's called apoptosis. That's the fancy word for it. But basically, they just lyse all their cells, take the nutrients away, and so they're trying to really deprive the fungus of any nutrients in that area. And then they also start to ramp up defenses, physical defenses in that area, where they tend to ramp up the cell wall of the cells right where the fungus is to try to almost like a moat around a castle idea. They're trying to limit physically the spread of the fungus by just beefing up their cell wall structure and integrity. And so when they see the hypersensitive response, it often comes right after this oxidative burst with the reactive oxygen species playing a role in that. And so they've tracked it. They often see a hydroxyl radical, that gets, the amount of that gets increased it spikes in concentration, or the superoxide ion or hydrogen peroxide. And so that is one thing that tends to happen. Then they've also documented nitric oxide is also being produced. And that one is being used as a second messenger. Second or secondary means the same thing. And what they're finding is that you need both the nitric oxide and the reactive oxygen species present in order for the full hypersensitive response to work. And so they think that the reactive oxygen species that are getting made can also damage the fungus, because any living organism does get impacted by reactive oxygen species, especially in high doses. Any organism will have a hard time with that. And so this idea that they're using the nitric oxide as a message, the ROS seem to be primarily as a way to kind of blast the fungus. And then they are trying to siphon the nutrients away from the fungus, kind of beef up the cell wall structure. And it can be fairly effective. Here are some studies where they stained the fungus. And so here you can see with the hypersensitive response, the fungus was limited to a small patch of cells. But on the right, that they made it so the plant couldn't do the hypersensitive response. And you can see how the fungus is migrating through the rest of the tissue and doesn't seem to be having any hard time with further colonization of the plant tissue. So that is one documented response that a plant will do, and it can be against a fungus, it can be against bacteria. And this is one of the earlier studies that was done. So they've known about the hypersensitive response for some time, but the nitric oxide having a role, that was a more recent discovery. So here you can see the hypersensitive response is kicking in, and they link it to production of nitric oxide. Okay, so that's where they filled that in just recently. So part of the hypersensitive response is that idea of a physical barrier. The plant does ramp up that part of its body to try to increase structural barrier, and that can be upregulating lignin, just putting more lignin into the cell wall, the secondary cell wall. It can be the formation of callus. We talked about callus way back when we looked at phloem tissue and how that can be a component to help plug the holes in the phloem. And then there's this process that I'm not going to go into, but they call it cell wall cross-linking. And basically when they cell wall cross-link, it just means they're strengthening the cell wall area and just making it a little bit tougher for the fungus or the bacteria to get through that. So the other idea that plants can do when they're getting attacked by a bacteria or fungus is that they can make their own enzymes that can try to attack that bacteria or fungus. And so those enzymes, the umbrella term for those enzymes are called PR proteins. Again, they like to give you jargon in this field of study. So when you hear PR proteins, those are just enzymes. They're just enzymes that the plant makes that is used to try to kill off the bacteria or the fungus. And so here, the second bullet, those are some examples of what those PR proteins could be. Maybe they're glucanases, chitinases, hydrolases. So chitinases, of course, are targeted more specifically for fungi. because fungi have chitin in their cell wall. Bacteria don't have chitin. But just trying to give you a couple of examples. So these PR proteins are enzymes, and they started calling them initially pathogenesis-related proteins. So that's where the PR protein term comes from. But again, they're just enzymes. They're enzymes that the plant is using to try to directly attack the fungus or the bacteria. So these PR proteins, enzymes. So the plant does have that ability to figure out, oh, it's a fungus attacking me, and then it'll start to upregulate its amount of chitinases and use the chitinase enzyme to try to break down the cell wall of the fungus. So here you see in the white box, they talk about lignin biosynthesis of hydrolytic enzymes. So that's what we're talking about in that previous slide there. Phytoalexins is on the list here as well. So phytoalexins are made in response to microbes that are attacking the plant. And it's a very rapid response and very effective response. So all the chemicals that they see the plant making when they test those in the lab, they tend to have really good, strong antimicrobial activity. And you can't like pin it down to a certain, like, oh, they're all alkaloids or they're all terpenes. You can't say that. They're diverse, and it really does depend. And so this phytoalexin term is just kind of a broad term, umbrella term again, talking about all these diverse chemicals that have really strong antimicrobial activity. And so they always say how these are regulated at the level of transcription. That's just a fancy way to say that the plant doesn't have these enzymes up and running on a day-to-day basis. All right, they didn't go through transcription and translation to make the protein. They have the genes necessary for it, but they won't turn those genes on and transcribe them until that bacteria or the fungus is starting to attack. And again, that makes sense when you think about all the different pathogens that plants are faced with on a day-to-day basis. It just does not make sense to have everything up and running. That would be exhausting for the plant. It would not be able to function. So wait until you're attacked and then start ramping up your phytoalexins. These are just a couple of examples of what they could be. There's lots that have been studied. This is a drop in the bucket compared to all the stuff that they know. But trying to show that there's some chemical diversity here. We talked about resveratrol back in the secondary metabolite slide set, and that one is also a phytoalexin. And it's found in red wine commonly. So phytoalexins can be quite potent and effective. So that's another strategy that plants can have. Then you see on this slide, over on the far right, they talk about systemic acquired resistance. So I'm going to talk about systemic acquired resistance, which they abbreviate down to SAR, and they say it that way, SAR, S-A-R. And then I'll be introducing another term that kind of sounds a lot like this one. So I'm going to put out the caution right now. Try to get these nailed down so you don't get them confused, because I'll be talking about systemic acquired resistance, asking you questions about that one, and then the other one that I'll introduce here in a bit. So what is it? Systemic, whole body. That's what that means. It's the whole plant body that we're thinking about, and acquired resistance. So it is something that allows for the whole plant body to be able to become resistant. They acquire the resistance. And so we've touched on this already when we looked at the idea of the caterpillar attacking one of the tomato leaves, and yet we see that the signal transduction pathway through the whole plant body and is telling all the leaves of the tomato plant, hey, get geared up. That's what we're talking about here. And so it's resistance through the whole plant body after pathogen attack in one part of the body. And so here in this drawing, they're showing on the lower leaf that there's something attacking maybe it's a fungus that's attacking that leaf. And the systemic response is that the signal is getting sent to all the other leaves and telling the whole plant to ramp up its defenses. They've also noted when they follow that experiment through and look at exposing those same plants to the same pathogen in a later date, so they'll hold off and then they'll re-expose the plant to the exact same pathogen, and they'll see that the plant does have elevated defenses. So it's analogous to our memory T cells that we have. When we get exposed to the same pathogen, we have those memory T cells coursing through our body, and they're ready to immediately defend. And so they do see that with plants as well, that plants can have that memory of being attacked, and they will have an elevated defensiveness. They can really do an effective job if they get attacked by the same thing at a later date. So kind of cool to see that comparison with our immune system. So when they tracked this down and looked at it more carefully of what is the signal transduction going on here, what they see is that salicylic acid hormone is used pretty much all the time. When they look for it, it's there. And so what they see is that on the leaf that's getting attacked, they'll see that salicylic acid synthesis and concentration is high at the site of attack. And then what they also see is that the methyl version of salicylic acid, so methyl salicylate, is a volatile form of that hormone, and that can get made and is used primarily to let the rest of the plant body know that they need to defend themselves. So it's kind of two versions of the hormone, one that's being produced locally, and then salicylic acid can also go through like the phloem, and then you have methyl salicylate that's volatile, and that can go through the phloem as well, and it can also fully volatilize out of the plant body and waft over to neighboring plants and tell those plants to go ahead and ramp up. And so it's a way for that signal to go out into the habitat. And if nearby plants can understand that signal, they'll start ramping up their defenses against that particular pathogen. Okay, so systemic acquired resistance. Here, we're showing that visually. Here's the pathogen on the lower left leaf, and they're trying to show how you have salicylic acid coursing through, methyl salicylic acid coursing through, and then they're trying to show by the red squiggly lines that's the volatile version coming off out of the stomata and getting released. And they're showing here that a neighboring plant then can pick up and hear that signal and respond appropriately by ramping up its defenses against that certain pathogen. So pretty effective. It's just a minor shift with that attaching the methyl group, and it's able to then get that signal much further out into the habitat. Okay, so that's systemic acquired, looking at the full body systemic, full body response to become hopefully resistant to the pathogen. Now, here's another topic, and this is different. So this is induced systemic resistance. Unfortunately, they sound very similar. So try to think of some kind of system that you can do to keep these two separate in your head, because this is a very different idea in the sense that it's actually beneficial bacteria that are kicking this off. So they've been called different things, depends on who's writing up the paper. They may be referred to as biocontrol agents. PGPR is plant growth promoting. The R stands for rhizobacteria. So the R is rhizo. That's supposed to be an O. rhizobacteria, and then PGPB, that's plant growth promoting bacteria. And then PGPM is plant growth promoting microbes. So unfortunately, it depends who's writing it, what the jargon is. But basically, we're looking at microbes that are beneficial to the plant. And they can be bacteria or fungi. They studied bacteria probably more heavily, but now they're realizing fungi can actually do this as well. So perhaps that should be amended to say beneficial bacteria and fungi, because they really have found both. So let me get rid of that. Come on. What we're finding is that this does not work the same way as the other one we just talked about with the systemic acquired resistance. So we don't see salicylic acid playing a role. So it's a different signal transduction pathway, which kind of makes sense if you figure out that it's a bacteria or fungus that's kicking this off. And so it also doesn't seem to have anything to do with upregulating the pathogenesis related genes, so all those genes that make the enzymes. It is a different suite of hormones involved. So we see jasmonate or jasmonic acid and ethylene. Those two are involved instead of salicylic acid and methyl salicylate. So it's a different pathway, which makes sense when you think about it, because how they get kicked off is a little different. When you looked at the other version with systemic acquired, it was a pathogen kicking off the response. Now we have the good guys kicking off the response. And so it's two different pathways. So here they're trying to show that idea where you have non-pathogenic microbes that are hanging out with the plant. And so we're talking about bacteria or fungi that coexist with the plant long term. All right, so they're living in close association. They might even be a root nodule. Here they're showing a root nodule, a legume, perhaps, where it's a nitrogen fixing bacteria, but it doesn't have to be that. They can just be normal microbes hanging out that are on the root, for example, and they're picking up the idea. The bacteria or fungus can sense that there's a pathogen, plant pathogen, and they essentially warn the plant through this signal transduction. And so it's a microbe telling the plant, hey, there's a dangerous microbe here, and you need to gear up because it's going to attack. And so it's kind of an interesting process where they're trying to show here that the bacteria is kicking off the signal and that you can get mobile movement of the hormones through the plant body. So it's still a systemic response. It's just that the good bacteria or fungi are the ones kicking it off instead of the pathogen, like with the other systemic acquired. Right, and they're showing jasmonate, ethylene. And so they've documented this pretty carefully. And when they first started doing the research in this, they weren't really hypothesizing that this could be possible. They thought that was pretty far out there, but they started looking and they did discover that there's actually many species of bacteria and fungi that do this, that they will warn the plant that there's a pathogen lurking, usually in the soil, that the good bacteria are picking up on, for example. So they're trying to show that you have these good bacteria, plant growth promoting rhizobacteria. So that's just the umbrella term for all the good guys, in this case, referring to the bacteria. And what they're trying to show here is that when they're hanging out with the plant, they can be directly trying to help the plant by producing antibiotics to reduce or kill off the plant pathogen. They've documented that really well. They've documented that these microbes actually talk to each other through this process that we call quorum sensing. And so these microbes are in the soil. They can actually chemically communicate with each other, and they can actually team up with each other to better fight off a pathogen, whether it's a plant pathogen or even a pathogen that's trying to attack them of some sort. And so they actually refer to the idea of cooperation, that through this process of quorum sensing, they're actually using that as a way to coordinate their behavior, all ramp up in this case to produce higher amounts of antibiotics that can kill off the plant pathogen. We also are recognizing that plants can kind of direct that process. So plants have a lot more control over what microbes are around them than we ever thought possible. So plants are actively recruiting and inhibiting certain microbes around them, and they have a lot more control over that than we initially thought. And so they are sending out their own chemicals that can actually communicate with these bacteria. So plants can quorum sense as well. They have the ability to do that where they can talk to the bacteria and fungi hanging out with them. And that conversation can go both ways. So it is back and forth. You can see here also they're showing that the plant can serve as a nutrient source by just the roots being so leaky. You get lots of chemicals being secreted out that the microbes will benefit from. So the plants, through that action, they are actively sending out certain key molecules to attract some bacteria and fungi and to keep others at bay. And then here you're seeing how the bacteria are enhancing plant defenses. So like that's the induced systemic resistance idea. So lots of things going on in this figure, but they have been, documenting this over and over and over. When they first documented it, it was kind of like, oh, that's probably just unique to that particular bacteria. But they realized that this is a common strategy. All kinds of plants and all kinds of microbes that hang out with them are doing that. So this is not from your book, but it's from a paper. And I just put this in here to, again, get you to think about the complexity going on here. This is an even more complex idea in the sense that it's a soil microbe, a beneficial microbe that actually tells the plant to close down its stomata, even though it's in the soil. And so it's in the soil, and what happens is you can read the step by step up here, but basically when this pathogen is on the leaf, the pathogen gains access through the stomata. And so what happens is the bacteria create a situation where they are telling the plant, you need to shut down your stomata. And it actually creates a situation of a single cascade flow, step by step by step. And the end result is that the plant closes down its stomata. You take away those bacteria and run the test again, and the plant's response to close down its stomata is significantly hampered. And so really nice study showing that here you have a soil bacteria telling the plant to, close down its stomata way up in its leaves. And then they're also showing on the same diagram the idea of plants being connected underground through mycorrhizae, and that chemical signaling can occur that way to help deal with plant pathogens as well. And so here they're showing a fungus attacking, and that the nearby plants are getting the signal that that's happening and they need to ramp up their own defenses. So very complicated. The more they look into this, the more complicated it's getting in terms of the interaction between these coexisting microbes in the plant. So that's the induced systemic resistance idea. And then I did also want to, allelopathy has been well documented for a long time, looking at how plants directly negatively impact other plants nearby. And so allelopathy is looking at plant-produced secondary metabolites. Often it's through the root system and mycorrhizal fungi that you get the passage of the chemicals into the soil. And it tends to make sure that neighboring plants don't really have a chance to grow. And so black walnut around here is pretty good at that. If you ever see black walnut in a forest system, they usually don't have a lot of neighbors really close by because they're good at this. Here is an example of spotted knapweed, which non-botany people, when they're out around in this area, and they, oh, look at those pretty purple flowers. And I'm like, oh, no, evil. And they're like, what? They don't know. So I had some popping up in my backyard, and oh boy, I went right at it because those things, once they gain a foothold, it is so challenging. And so it is a monoculture. And yes, those are purple pretty flowers. I'll grant you that, but they're an evil, evil plant. So if you see them, and if you are so inclined, go ahead and yank it out of the soil wherever you see them. Down by the Jacob Eddy building that they're redoing right now, there's a lot of construction now, but by the hoop house down there, we have the field plot, and it was just covered in spotted knapweed. So that I was worried that that was going to interfere with the experiment that one of my grad students did. So we had to test for I think it's catechin is the chemical that it produces. So we had to run a test to see the levels of catechin in the soil, because that might impact. Turns out it doesn't seem like it did impact, thankfully. So in its native habitat, on the left here, they're showing that you see one flower showing up in a native patch over in Europe. So plants over there know how to deal and interact with spotted knapweed, and it's kept in check. Over here, the plants don't have that ability. It's an invasive plant, and so it's easy to get in and it's been established. And now they're showing that once you have spotted knapweed as a monoculture like that, it shifts the mycorrhizal fungi who's there in the soil. So some of you may have had summer jobs where that was your job, to pull weeds. I know many students, that's what they do for their summer job. They get jobs all day long, go to different field sites and pull spotted knapweed. Unfortunately, so the idea there is pull all the spotted knapweed out and then replant that area with native plants. And they're having problems with that. And they're like, why won't the native plants reestablish what's going on? And so they went into the soil. A microbiologist went into the soil and said, there's probably something going on. And they realized the mycorrhizal fungi that were there initially hanging out with the native plants disappeared or nearly disappeared. So their levels have crashed because the spotted knapweed doesn't interact with those mycorrhizal fungi. And probably was directly inhibiting them. So it's challenging. If you get the spotted knapweed out, that doesn't guarantee you're going to get your natives to reestablish. They need to really think about that. So, oh yeah, there's the catechin chemical down there. So pretty successful plant. In that video link that I posted in EDUCAT, what plants talk about, they do talk about, I think they talk about spotted knapweed out west and how it's causing a problem. So other plants behaving badly here, we're seeing some parasitic plants essentially. So here, figure A, you're looking at the mistletoe plant, which is appropriate because that's kind of a thing around the Christmas holidays. And so you can see how it's, this green bushy like plant that likes to hang out on the branches of trees. And then dodder, this orangey plant, they're showing on a sand dune area, that's dodder. And then a close-up of it, they talk about that one too in that What Plants Talk About documentaries. So very interesting plant. The dodder plant needs to germinate and grow and find a host all within 24 hours. If it doesn't find a host to plug into, it dies. So it's an amazing plant in how it can be so successful with that kind of dire restrictions put on it. So that plant can actually, in the video, you'll see how the plant can actually pick up on volatiles around it and know where it's coming from and plug into the plant of choice. And so it has favorite plants that it likes to parasitize over others. So a really interesting study that they did on that. Mistletoe is an interesting plant. It is called a hemiparasite, H-E-M-I, hemiparasite, which means halfway essentially. And so this is kind of like the worst parasite of all because it could do photosynthesis on its own if it wanted to. It just chooses not to. It would rather parasitize. And so I listened to a podcast on, there's people who actually hunt mistletoe for a living. And I like, you get mistletoe sprigs around Christmas time. You're supposed to kiss under the mistletoe. But it's like, how do they harvest that? Because I knew it grew up on branches. I'm like, well, how do they can't exactly drag ladder into a forest? So what do they do? And so that podcast, they actually use rifles and shoot them down. Have you heard this? I'm like, that is awesome. So they go and it's usually in the southern states that mistletoe is present enough to make money off of it. But yeah, they go into the woods. I'm like, do you need a permit for that? Or I don't know the details, but they actually shoot it down. And they were talking to a father-son team that's their winter business. They'll go in there and they'll shoot. And he was getting all into it. He's like, yeah, you have to be really careful where you aim because you don't want to obliterate the bundle. You get more money if it's all bundled. And so you want like the whole thing to come down. And so I'm like, wow, okay, now I know where all this mistletoe is coming from. Who knew? So, but when you think about it, it's not like you can drag a ladder around to go and clip that off. That would just wouldn't work. So
some people like climb trees for.
Yeah, but again, that would be really long. It's much quicker if you're a good shot. You have to be a good shot, but it'd be a lot quicker just to shoot it down. So, and then the other question is, how does it get up there? Has anyone seen that video? I show it in some of my Plant Kingdom classes, but if I can find the video, they take it off YouTube a lot. I don't know why, but there's a video. It's a Sir David Attenborough video clip. Yes. So I can't remember the bird. It's a really colorful bird with a red breast, and they show it eating the mistletoe fruit, and it's a really sticky fruit. So the bird knows how to like squeeze the fruit, get the seed to pop out. It eats the seed, but it stays sticky all the way out the rear end of the bird. And so the seed is coming out the rear end of the bird, and you can tell the bird doesn't like it. So it's like, God, what is this? Dangling out of his butt. And so they show the bird wiping his butt along the branch. He just kind of moves along the branch and he's wiping his butt, and finally the seed comes off. Makes me think I had to pull some. My dog went to the bathroom and didn't. Long hair was stuck to it. I'm guessing it was mine. I had to pull it out. But anyways, that was fun. I had to do that right around midnight last night. It's like, oh, good night. So, but then the seed sticks right onto the branch. And then they do the videography where they show the root radical coming out and plugging right into the branch and starting to plug into the phloem and siphon off the sugar. So extremely successful plant. And like I said, it could do photosynthesis on its own. It just chooses not to. So here is how it's plugging into the branch. So again, the word haustorium is just a general term that they use for any kind of what is a term they use for really aggressive growth into tissue. In this case, it's the mistletoe radical docking in and getting at the stem.
So does the plant like try to defend?
Yeah, I haven't read much on how a plant deals with mistletoe. Usually it doesn't kill the plant. The mistletoe doesn't kill the host plant because you can see mistletoe in Australia. Not that I've been there in person, but I've seen pictures of plants in Australia with mistletoe all over in the crown of the plant, and the plant looks fine. So I think if the plant got stressed with something else, it might, be difficult. But in general, it doesn't seem to harm the plant. Now, this, on the other hand, I don't know what a plant does. That's not Photoshop. That's like for real. So it's like, what do you do with that? I saw that, just laughed. It's like, okay. But apparently, and I confirm this, I know people who own goats, and they're like, oh yeah, goats will do that. They're that good at climbing, hanging onto the plants. So it's like, yeah, like what does that poor plant do? I'm sure it's trying to, pump out secondary metabolites into its leaves, but look at that poor plant. That tree is just denuded. It's gone. Yeah. So anyways, I don't know what the plant must be trying, but it didn't work. So those crafty little goats. So anyways, the last few slides are just clippings of screenshots of different publications in this area of how plants are defending themselves, how are they communicating, and getting at how are even cooperating. So that video that I posted that I hope you'll watch over break will touch on some of these ideas. And it's really opened up a really neat field of research looking at how plants are, they behave in very particular ways, and they can sense what's going on around them in a very sophisticated way, in many sophisticated ways. And so there's actual, books, Plant Behavior and Intelligence. All right. And so it depends on how you define that, of course. And they always define it. They'll say, this is how we are defining it. And then they walk through the data and the evidence to support that idea. So some good books to think about if you wanted to investigate this a little bit more. Brilliant Green, I've read that one. It's an easy read. And it was only $3 when I checked, when I posted that. So not very expensive. And this one was under $10, What a Plant Knows. You may have heard of The Hidden Life of Trees. I talk about that in Plant Kingdom. Sometimes assign chapters of that book as well in that class, because I think he does a good job of getting you to think about plants from a different perspective that I think is relevant. So anyways, some things to think about or read when you get a chance to actually read something for fun. I know that's not going to happen anytime soon, but maybe in the summer. And then I've had students in the past, they say, okay, so all these secondary metabolites, I have said to you more than once during this section that, for us, that's good nutrition. If we eat plants that have these secondary metabolites, that's considered healthy. And it's like, well, why? Why is it? And so this slide is for anyone that is asking that question. And basically, if, how many times has your mom said, eat your broccoli? It's got good antioxidants in it. And the idea is that it's not really, they have enough of the antioxidants for your nutrition, that broccoli doesn't make enough antioxidants. So a serving of broccoli won't get you all of your antioxidants. But what it does is it produces a mild adaptive stress response in our cells. And so what the end result is that it gets our cells to start ramping up our own antioxidant enzymes. And so it's kind of this indirect impact of why it's good for you. Okay. So broccoli does have antioxidants, but at the level you could get from your diet, they're not going to function. There's not enough antioxidants in that broccoli to be of full use to you. It helps, but it's not like the full thing that you need. And so they're saying those secondary metabolites that we're eating is actually causing a mild adaptive stress response in our physiology. And that's what's causing the healthful result of that. Okay. I just thought I'd put that in there if anyone's interested. All right. So we finished that topic, that slide set. So on Friday, we'll get into abiotic stress. We'll look a little bit at abiotic stress on Friday.
So like the